Okay, I have a question from Anya that says, when you prepare your sample for H&E, what is the main things to consider? So if we're talking about wax embedding samples, of course, first of all, you can't start staining your samples directly after you cut or section them. So they have to have properly dried on the slide. Otherwise, you will see if you are too quick after cutting the sample and you start staining it, your sections will most probably slide off while you're staining it. So that's the first thing, is just wait a few hours before you start actually sectioning your sample. But for the others, I think depending on the tissue that you're staining, the protocol for each lab will differ, and each tissue sometimes require a bit longer in certain chemicals, but as well, just a proper dewaxing, proper cleaning, and then having a good amount of time in the actual hematoxylin and eusin. And then also, of course, afterwards cleaning it properly so you don't have any background stains as well. So depending on what sample you do, and if it's wax or maybe cryo, it will depend on a few things that you have to consider. Another question is that ask if I have experience in histology done on insects. Not histology itself. I've done a few studies with students that viewed the insects with the scanning electron microscope, and some also with the confocal, where they actually, for the SEM, for example, they viewed the surface of the insects. And another type of study also looked at the different characteristics of the insects to be able to classify them in their families and groups. And the other one that looked at the surfaces of the insects looked at why the insect has the property to actually be able to float on water. And that gave very, very impressive SEM images, and this detailed structure on those insects is actually very fascinating. Depending on how big the insects are, maybe you will section them. That will probably be to actually see the internal structures, not necessarily the outer structures. So yeah. But they are very interesting little things if you do view them with the same. See another question. So the question is histology acceptable to algal cells too? For cell cultures, there are a few different techniques that you can do, and it doesn't necessarily involved in preparing your samples for or going through the wax embedding or cells cryo embedding. Sometimes you can view your cells live, and then with if you have the correct microscope, you can use then the phase contrast of the microscope to be able to actually view the sample itself. So for that cases, sometimes you don't then need to stain the cells, but with your phase contrast objectives that are on the microscope, you will still get a good contrast to view your cells. So, but yeah, because you will only end up with a very thin section or a thin layer of cells, it might be, or it will be more tricky to section because you only have then a few micrometers to work with. So I hope I answer your question. Then there's another one from Anya. So the question from Anya is, how do you decide between using a confocal and a normal fluorescent microscope? So when is a normal fluorescent microscope enough for your research, and when do you need a confocal? So fluorescent microscopy is a good, I would say, way to start. It will give you plenty of information. Confocal, of course, you are, because of its resolution that you can also get with confocal, and with the lasers, I would say if you're looking at very small proteins or structures in the tissue, tissue or cells, I would suggest using confocal. But sometimes the normal fluorescent microscope is more than enough to view most of the samples. And with today's software that's out there, the type of fluorescent images that you can get is very close to confocal images, not the high-end confocal, but you can get quite good fluorescent images from the microscopes, and you don't need that looks similar to confocal images, so you don't have to use a confocal for that. Yes. Any other questions? Yeah, if you don't want to type the question, you are more than welcome to switch on your mic and ask the question yourself. So you're more than welcome to do that as well. So the one thing also to remember is that sample preparation for me is the most important part of the study. You can change some of your results, I would say, very easily if you do not prep your sample correctly. That is, for example, if you keep your samples too long in a fixative or a certain step, then your cellular structure at the end of the day might differ from what you would have seen at the beginning. So remember that the main thing that you want to do with sample preparation is to make sure that at the end of the day, when you view your sample under the microscope, is that it looks as close as possible to its native state. So meaning, if you were able to take your sample and put it directly under the microscope, does it look the same? Or that's what you want to see after you've went through the wax embedding or any sample preparation. So you don't want to distort structures or break them or to damage them. So that's the most important thing. Although it's a very, very long and boring process for most of people, it is for me the most important part of your sample prep work. And like I said, if not done correctly, it can cause a lot of problems at the end of the day. But of course, that is also why your study has to include controls. So if there is some damage that you caused, you will then know it by looking at your control. Okay, then we have a question here. So if you want to monitor your cells in terms of if you want to see them growing, for example, then yes, live cell imaging, I would suggest using confocal. The reason is, I know in Stellenbosch University, the confocal has an incubator chamber on, meaning that you can still control the CO2 and the temperature inside the microscope. So you can put on your flask and you can leave your cells still happy inside or on the microscope. So yes, in that case, I will use a confocal. And also, even depending on if you're going to stain your cells or just view them as is, this confocal at Stellenbosch can also do that. So maybe some fluorescent microscopes with then this little case around them that can monitor your CO2 and temperature you can also use. So here in Stellenbosch, I would suggest using your confocal or the confocal, but if you are at a facility that maybe has a fluorescent microscope with one of these incubated cases around them, then that will also be something that you can use. So yeah. Cool. So I think we're going to have another 4 minutes. If anybody has any questions, please do ask. I did put or you should have my email address as well. So if you think of something afterwards, you are more than welcome to email me and I will answer your questions as well. But like I said, even you can always ask someone like Lisa from a CAF unit or any microscopy unit about these questions. The people working at microscopy units do know a lot about preparation and what will be the best for your sample. So you know they're always a good starting point, especially if it's a new sample or new cells or just a new area that you are moving into and not sure what to do. Because remember, I think most of you will know that antibodies are quite expensive. So you don't want to buy just because you think it's the correct one. You please go and ask an expert in that field to guide you to what antibody to buy or stain or whatever, so that they can guide you into what you need to buy as well. Because of course antibodies can cost up to 10,000 Rand. So in today's economy, you don't want to waste that funding on something that you might not use or buy the incorrect one, because antibodies and primary and secondary antibodies can be quite tricky for a newcomer to the process. So please do ask someone that knows what's going on before you buy them. You don't want to waste your money on buying incorrect antibodies. Okay, I think if there's no other questions, I think we will end this session. So like I said, you're more than welcome to email me, or if you're in the area, you're welcome to come to my office. We are here a few days of the week now with lockdown. So you're welcome to make an appointment to come and see me if you have another questions as well. So yes. Okay, we have a question about food samples. What specifically do you want to know about them? What type of microscope do you need to use? Do you need to stain them? What do you actually want to see with your samples as well?
You can unmute your yeah. That should maybe go a little bit quicker than typing.
Yes. Yeah, you can switch on your mic and ask the question, so you don't have to type.
Hi, can you hear
Hi.
me?
Yes, I can hear you.
Okay. Actually, I'm working on food samples, legumes and cereals. I'll be making cookies.
Mm-hmm.
I'll fortify those my maize with legumes. That's bambara, and I will feed lab rats. So will I still be needing the microscope experience to work on it?
So you're gonna do an animal study and then feed them the different cereals to see the effects of them.
Yes, actually, this is my first time going through this study, so I just want to know, am I going to see any like changes? Because I'm just being told I'm going to be feeding those lab rats, So I'm just still getting more knowledge because I don't think.
Yeah, so it depends on the organs that you will harvest from the animals. So of course, if you're looking at diets and the effects of that, you might consider maybe if you want to look at insulin levels or something like that, you will take the pancreas. The liver is always a good tissue to look at. Maybe the GIT, it depends. So yes, I mean, then you will do normal. You can do normal H&E or wax embedding and then H&E staining to see if there's an effect. Of course, if you are looking more for something on insulin or levels or something like that, of course you can test the blood for that levels. But you can also then maybe get a specific antibody to look at if there's an increase in insulin in the pancreas itself as well. So that's something that you can also do, but that will depend on if that is something that you're interested in your study.
Well, sure, it is.
Yes, yeah. So I mean, so there's if insulin testing can be done in a few ways. So I mean, you can, like I said, do the blood testing. That's a nice, I would say, easy way to do it, because it's not involves a lot of preparation. I mean, normally people send that type of blood to pathology labs to get tested for them. But if you want some histology in your in your thesis or research, then taking the pancreas will then be crucial. And then you can either just do normal H&E or H&E and then doing fluorescence or immunohistochemistry as well.
Okay.
Okay.
I'll take note of that. Yeah, thank you.
Cool. Good luck.
Thank you.
Okay. So if there's no other questions, I will end this session. Thank you for everybody that participated or were on the meeting. And then, like I said, any other questions, you're more than welcome to email me. And just keep in mind to always ask people about your research question, or keep your research question in mind when you are going to talk to people of the microscopy unit, so that you save yourself time as well as money when you do your research itself. Okay, so guys, thank you, and then stay safe, and hope you have a wonderful day further. Goodbye.
Thanks, Chantal. Bye.
Bye.